All right. Well, hi everyone. Thank you for joining us tonight. My name is Rowena and I'm the Bushland Community Liaison Officer at Willoughby City Council. So we do have a very special guest speaker joining us tonight. But before we start the presentation, I would like to begin with an acknowledgement of country. So on behalf of Willoughby City Council, I wish to acknowledge the traditional inhabitants of the land on which we stand, the Aboriginal people, their spirits and ancestors. We acknowledge the vital contribution that Indigenous people and cultures have made and still make to the nation that we share today. So today's topic revolves around discovering and connecting with nature. Obviously, connecting with nature is becoming more and more important, especially since the pandemic. More people are now learning the importance of the natural world to both our physical and mental health, not to mention the many pressing environmental issues that are threatening our natural world. And Willoughby City Council and its bushland team in particular are dedicated to caring for and protecting our natural environments and all the flora, fauna and fungi that depend on it. So we hope this presentation tonight will inspire you to go outside and discover all the wonderful things that nature can offer. And one of the ways that people can connect with nature and even contribute to its conservation is by becoming a citizen scientist and collecting really valuable biodiversity data through platforms such as iNaturalist. So some of you may be familiar with this platform, or maybe this is completely new to you. So I'm very excited to introduce you to our speaker tonight, Thomas Magzaglio. Thomas is a PhD student at the University of New South Wales. His research focuses on species discovery in and knowledge of Australian plants. So Thomas frequently uses the iNaturalist platform and the data to answer his research questions, having worked on a wide range of projects from ecosystem and bushfire recovery, marine forensic science, and invertebrate taxonomy. Thomas is a curator and a forum moderator on the iNaturalist platform. Just to give you an idea, he has made over 37,000 observations on iNaturalist, as well as over 200,000 identifications made for other users. So it's very impressive, I must say. He is also the organizer for the upcoming Great Southern BioBlitz for Sydney, which I'm sure he will talk more about later on as well. So if you have any questions, please pop them in the chat and I will direct them to Thomas at the end of his talk. So without further ado, I will pass it on to you now, Thomas. Thank you.
Thanks a lot, Rowena. So pretty much just the way I'll run things today is I'll have a brief presentation just on what iNaturalist is, some of its functionality and what BioBlitzes are in general, as well as the Great Southern BioBlitz specifically. And then I'll actually use iNaturalist on the screen and actually show you how to upload observations, which is mostly what you'll be doing during the BioBlitz. And then of course, plenty of time at the end for questions if people have them. The one thing I did want to mention before I started is that all of the functionality and the tutorials that I'm going through this evening all relates to the desktop version of iNaturalist. So you can use iNaturalist on your computer, but you can also download it as an app on your phone. And the reason I always like to bring this up is that although the app on your phone is very convenient, and it is useful if you're out in the field and you're on the go and you want to snap a few quick photos and upload them while you're in the field. But the desktop version of iNaturalist has far, far more functionality than the phone apps, especially if you use an iPhone. The iPhone iNaturalist app is quite bare bones compared to the Android app. But then even the Android app has greatly reduced functionality versus if you're on your desktop. So I do certainly if you've taken photographs with your camera, or even if you've taken photographs on your phone, I would recommend waiting until you get home and uploading those photographs via your computer. And it's just a lot easier for you and a lot more of an efficient process. So what I'm going to do is I'm going to go through my brief presentation, and this will just be largely a broad outline of what iNaturalist actually is. So what is iNaturalist? So it is an online biodiversity citizen science platform. And it's one of the biggest such platforms in the world, if not the biggest, in terms of the number of users and the number of observations that get uploaded to the site. So what I mean in terms of uploading observations, so iNaturalist runs basically on two very simple principles, observations and identifications. So any user from around the world can go out and take photographs or sound recordings of any organism they encounter. So this can be anything from birds to plants to insects to fungi, even things like bacteria or viruses. If it's something that's alive, then it's fair game. Once you've taken those photographs, you then upload them to iNaturalist, again, either on your phone app or on your computer. And the really amazing thing about the site, and one of its great strengths, especially as this collaborative platform, is that you have this tremendous community of other enthusiastic naturalists and experts from all around the globe. So there's over 2.5 million users that use iNaturalist right now. And not only do you have many enthusiastic amateurs, but you also have all kinds of amazing experts that are so happy to share their knowledge. So you have museum researchers, curators, PhD students, taxonomists, and even for many groups of organisms, literally the world experts in those groups that use iNaturalist and are able to offer up their expertise and knowledge. So once you've put up your photographs or your sound recordings on the platform, either you can make identifications if you already knew what you saw, or other users can identify your observations for you. And then one of the best bits about the platform is that your data then gets piped to external databases. So from an Australian perspective, your data will go into the Atlas of Living Australia. For people that are unfamiliar with that platform, it's basically a massive biodiversity data aggregator. So it's a repository of data that anyone can freely access and use online. And it basically accumulates data from all kinds of different sources, one of which is iNaturalist. So other data sources that feed into the ALA is things like Frog ID that a lot of you might have heard of, or eBird, or more government-based things like Bionet. And then on top of that, a subset of that data also goes into the Global Biodiversity Information Facility. And that's basically exactly like the Atlas of Living Australia, but at a global scale. So it's pulling in all kinds of biodiversity data records from across the entire planet. To give you a bit of a sense of scale, the Atlas of Living Australia has, last time I checked, somewhere in the vicinity of about 115 million data points, whilst the Global Biodiversity Information Facility has about 1.5 billion. So once your data is in these databases, then it actually gets used by researchers in real life scientific research, which I always find particularly gratifying to know that, the photo that I took of this bird or this plant or this bug might end up being a really useful data point for actual research that has a meaningful impact in something like ecology or even conservation. So just to give you a brief sense of what I was talking about, of the incredible magnitude of the community of iNaturalist, as of earlier this morning, we were almost at 119 million observations that have been submitted for the platform across the world. And we are getting very, very close to that 400,000 species observed mark. You can see that almost 2.5 million people have contributed records. And then if we look at things from an Australian perspective, Australia is one of the heavyweights on iNaturalist. The number one contributing country is, as you might expect, the US. But that was where iNaturalist was founded. And of course, they have a much bigger population. But Australia really punches above their weight, and we're up in the top 4 contributing countries. So that's the US, Canada, Mexico, and then Australia. And then you have other countries like Great Britain, South Africa, Italy, and Germany also coming in the top 10. So you can see with Australia, in the past month or so, we actually passed 4 million observations. We're getting close to 50,000 species, and we have more than 50,000 people who have contributed records across the country. And if we look at those statistics from a spatial perspective, you can see from this nice heat map that I made for a recent paper I put out on iNaturalist. You can see it's kind of what you would imagine, right? So all of the hotspots, which are in red, they're mostly clustered around the biggest population centres. So you can see Adelaide, Perth, Melbourne, Sydney, Brisbane. This one's up around Townsville and Cairns, down here in Hobart. So you can see generally most observations tend to get submitted where people live, which makes sense. But what I always find really exciting about this figure in particular as well is all of these blank spots. So if you do find yourself going on a holiday or a trip and you find yourself passing through the Red Centre or the Nullarbor or areas in Eastern Northern Territory, certainly make sure you try and upload some iNaturalist observations, and it's really helpful to fill in those data gaps. Another figure that I like to show people as well is just the incredible exponential rise in the use of iNaturalist. So what this figure is showing is this is specifically for Australia, and it's showing the number of observations submitted every month. So you can see back here when iNaturalist started and was in its infancy back in 2008 in the US, really nothing was going on from an Australian perspective. We start creeping along the years, and you can start to see there's a tiny little line here for 2013, 14, 15. But it wasn't really into about mid-2016 that things started to kick off, and then we never looked back. We've had exponentially increasing contributions to iNaturalist. And indeed, if you were to make this figure for number of observations, number of users, number of species, you get the exact same shaped curve. And it was about last year that we actually saw a number of observations exceed 100,000 for an individual month. And then late in last year, we had observations exceed 175,000 in a single month. And I suspect that in a couple of weeks' time, we'll actually have observations exceed 200,000 in a single month. And the reason for these big spikes is the Great Southern BioBlitz. So this is what you're all here for. This is the event that you'll be participating in just a couple of weeks. So essentially, the way that this came about was that there's been this event on for a number of years called the City Nature Challenge. And this is basically a 4-day event where people from cities all around the world go out and compete and see how many species they can observe within their cities. So some BioBlitzers participants enter as individuals, but in this case, you would enter as part of a city. So you would have places like in Australia, Sydney, Melbourne, Brisbane, Geelong, Adelaide, and basically you'd be seeing how many observations you can make in your city in those 4 days. But the problem with that BioBlitz is that it runs at the end of April and the start of May. So it's perfect for the Northern Hemisphere, but for the Southern Hemisphere, this is getting towards the end of autumn. Temperatures are getting a bit colder. Most things have stopped flowering. There's not many insects and birds around. So a few years ago, myself and the other Australian organisers for the City Nature Challenge, we thought, why don't we make an equivalent event, but one that is at a good time for the Southern Hemisphere? And so that's how we came up with the Great Southern BioBlitz. So we basically thought we'll have the City Nature Challenge, but just for Southern Hemisphere countries, and we'll have it running during our spring so that we get to make a lot more observations of insects and flowering plants and so on. And you can see last year we had this amazing turnout. So in just the second year of the event, we had almost 200,000 observations submitted across just 4 days. And you can see over 22,000 species and just really amazing participation as well with more than 6,000 people. So hopefully those numbers will increase even further this time around. But essentially the way the BioBlitz works is that you'll have the 4 days, the 28th, 29th, 30th, and 31st of October. And as long as you take photographs of any living organism anywhere in the Greater Sydney area during those 4 days, and you upload them to iNaturalist, they'll get automatically collected by the project and automatically counted towards Greater Sydney's effort in the BioBlitz. So nothing extra that you have to do, literally just take photos, uploaded them to the platform, and they'll all get collected for you. So really simple process. So what I might do now is I'll actually run through, apologies. I'll actually run through sharing my screen again and actually running through iNaturalist and showing you how to upload observations. So just to reiterate what I said earlier, this is showing you how to upload observations via the desktop. So this is your home screen that you'll see when you log into iNaturalist. And what you want to do is go to this big green upload button at the top right-hand corner of the screen. So you just click that, and from here you'll have two different options for how you can load your photos in. One of them is you can click the big blue Choose Files button, and then that will give you a pop-up screen that then navigates you to your Explorer Windows page. And you can just highlight whatever photos you want to load in, and they all get loaded in for you. Alternatively, you can actually just manually drag your photos onto the screen. So if I was to remove all of those, so if you just have Windows Explorer open up separately, you can go down, you can select all the photos that you want, and then you can just pick them up and then drag them onto your upload screen as well. And it works exactly the same. So once all your photos, I might just remove those again and then just add them via my other method. They're just ordered a lot more nicely when I do it this way. So once all your photos are loaded onto this screen, you'll notice that for each individual photo, it shows 3 important pieces of metadata. And I'll get back to those in a second. But one of the most important things when you're uploading observations to iNaturalist is that there's 2 general rules. So the first is that if you have multiple photographs of the same species or the same individual, it's really important that all of those photographs get combined into the same observation. So it's definitely recommended to take, 5, 10, even 15 photographs of the same thing if that's what's needed to identify it. But if you do take 10 photos of the one bird or the one plant, then those all need to be combined as part of the same observation. And then the flip side of that coin is that if you take photographs of multiple different organisms and different species, then they should all be uploaded as separate observations. So if you take photographs of 5 different plants, then rather than combining them all into one, they should be uploaded as 5 different observations. And so at the moment, you can see here on the screen, I've got 13 different photographs, but at the moment, they're all 13 separate observations. But obviously, some of these photographs belong to the same organism. So what I have to do is I combine them. And this is really easy as well. First, all I have to do is pick what I want the first photograph to be. Usually this is my best photograph or the clearest or the one that I think looks the nicest. So in the case of this snail, I'll say, okay, I'll pick this photograph as the first one. All I then have to do is pick up the other photos and then drag them onto that other photo. So pick it up, drag it on, pick it up again, and drag it on. And you'll now see that it says underneath 1 out of 3. And I can just page through the photos and you can see that they're all combined into the same observation. So I can then go ahead and do that for the other photographs as well. And you can see really easy process. I've now gone from 13 observations, I should say, down to 5 observations with the 13 photos distributed across them. Apologies, I'm just going to blow my nose. My hay fever is dreadful now with all this. It's dreadful, isn't it? All my research is based on plants, yet I get very bad hay fever. It's not the best combination. So you'll notice that all of my observations here, my 5 observations, they have 3 important pieces of metadata. Some of them filled out, some of them blank. So you'll see here that each one has a bar that says species name, each one has a bar that has the date and the time, and each one has a bar that has the location. And it's really important that for each observation that you upload, all 3 of these pieces of information are filled out. So first for the date. 99% of the time, if you upload a photo, no matter whether you've taken it with your phone or your camera, that date and time is going to be filled out already for you. If for whatever reason that isn't filled out, so that might be, for example, if you have uploaded a photograph to Facebook to show someone in the chat, and then you accidentally deleted it off your phone, and then you re-downloaded the photo from Facebook to then upload to iNaturalist, sometimes that does strip the metadata from your photo. But whatever the case, if for some reason the date isn't there, it's really easy to fill it in. You just click on that bar, you'll have a calendar pop up, and then you can just easily navigate to whenever you know you took that photo. So in this case, I know that this was July 16th. You'll notice that the time then gets filled out as whatever the time is right now. If that time is incorrect, then you can just delete it, and that's totally fine. As long as the date is there, that's what's important. You don't have to have the time as well. Now for the location, it's exactly the same concept. It's really easy to fill out, but in this case, the location is more often blank than the date is. So if you take a photograph with your phone and you have location services on, then unless there was some weird glitch with the GPS, most of the time the location should be filled out for you. But if you take photographs with your camera and you don't have a geotagger on, then that location will be blank. So in order to fill it out, exactly like the date, you just click on the location bar and you basically get given a pop-up window with a Google Maps screen. So you can do this in one of two ways. You can either just search for the name of the location, just like you would on Google Maps, or you can physically move the map. So if the spot you're looking for is already close by, then you can drag the map around and easily then put a point on the map. So in this case, I saw this up at Overlander in remote Western Australia. That is not where I saw that. So let's go for Hamlin Bay. Okay, well, it looks like there's 2 Hamlin Bays in WA. So let's just manually navigate. So yeah, so when you're typing in names, the names that you know for locations might not know, it's Hamlin Pool, that's why. So this is where I saw this snail up here in remote Western Australia, right here at this crossroads. So I can just click on the map and my point has been entered. Now you'll see that around the point where I clicked on the map, a red circle appears with little white handles. This is the accuracy circle. And what this basically, and you can grab the edges, you can grab the handles and you can make it as big or as small as you want. And basically the function of that circle is you saying, I don't remember exactly where I saw this organism, but I know that I definitely saw it somewhere inside this circle. So it's just a really useful way of indicating uncertainty if you weren't 100% sure where you saw that organism. It's always best to try and make it as small as you possibly can and obviously have as little error as possible. So once you've clicked on that point on the map and you're happy with your circle, all you have to do is click the blue Update Observations button and you'll see I now have the location filled out. One hurdle I often see new users trip over is that they'll enter the location just as a name. So the most important part of the location is the actual coordinates. So some people will actually go to the bottom here and type in something like Hamlin's Pool. They'll say that's where I saw it. Then they'll update observations and you'll see that the name does fill in there, but there's still no point on the map. And you can see the coordinates here are blank. Whereas you can see as soon as I click on the map anywhere, it actually fills out the latitude and longitude. So always just double check that with your location, you don't just have a written description of where you saw that organism, but you actually have coordinates filled in as well. That's the most important part. And then the final part of each observation is the species name. Now you can go about this in a few different ways. So obviously, if you've gone out and you've taken a photo of something like a magpie or an Australian white ibis, and you already know what it is, then that's absolutely fine. And you can just type in the name. So for example, this is a photograph I took of a juvenile Pacific gull.
Upstairs on the balcony. Yeah, beautiful bay.
Would you be able to just mute yourself if that's okay? All right, excellent. So in this case, this is a photograph I took of a juvenile Pacific gull. In this case, I know exactly what I photographed, so I can just go ahead and I can type in the name. Now, you'll notice when I start typing in the name that you'll have a drop-down menu will start to appear. And so this is basically like an autocomplete menu that you would see in any kind of search bar. And once the option appears that you want, just make sure that you actually do click that option. So it's not just enough to just type in Pacific gull in that case. You have to type in Pacific gull and then click the drop-down option as well. Now, let's say that I know roughly what something is, but I don't know what the species is. That is also completely fine. So let's say I have this plant here, and I know that it's in the genus Oxalis, but I'm not 100% sure what species it is. Again, that's totally fine. You can just type in Oxalis and you can enter that as an ID. So in this case, I can type in Oxalis, that option comes up, and I've just added a coarser ID than the previous observation. You can do this at any level. Let's say I took a photo of this bird and I had no idea what it was. I just knew it was a bird. Then I can go ahead and I can type in birds. Now, it is always better to put a coarse ID, even if that ID is just something like plant or animal or fungus. That is always better than leaving that bar blank. And the reason for that is that many experts and identifiers on iNaturalist specifically put in search filters when they're looking to identify observations for other people. So if you're a bird expert, then you are more likely than not go to all observations and then specifically filter to birds. So if you took a photograph of a bird, but you haven't identified it as a bird, then that person won't see your observation in their search and they won't be able to identify it for you. So even if I had no idea what this snail was, I'd at least type in mollusk, and then there'll be plenty of experts who filter to mollusks specifically, and they'll be able to find it and to identify it for you. Now, there's one last option for when you're filling out these bars, and that is with the computer vision. So iNaturalist has this amazing software called Computer Vision, and it basically functions, if anyone has ever used Google Lens or a similar application, it functions in pretty much exactly the same way. You present the software with a photograph, and then it tries to guess what species is in the photograph, essentially. Now, you always have to take the options that it presents with a bit of a grain of salt, depending on what kind of species you observed. If you took a photograph of a bird or a tropical fish or a large mammal or some kind of common plant, then most of the time, it will be able to correctly guess the species, and it's really, really fantastic. But then once you start dealing with more obscure species, things like tiny spiders or rare land snails or obscure plants from the rainforest, then it starts to struggle and it won't be able to guess correctly. And one of the reasons for that is that for a particular species to be guessed by the computer vision, there have to already be 100 observations of it on iNaturalist to then enter the training data set. So if you have a pretty rare species where there might only be a handful of observations that have been uploaded, then it is impossible for the computer vision to guess what that species is. But the problem, essentially, is that it won't just say, well, I can't guess. It'll still make a guess, but obviously the guess will be wrong. And that the guesses it'll present will be things that look similar, but obviously are not that species. And so this can be a bit of a trap that a lot of new users fall into. They'll kind of not use the computer vision critically enough, and they'll just accept any of the options that get proposed to them. And those inevitably end up being some kind of species only found on the other side of the planet. So if you do use the computer vision, that's completely fine. But it's always good just to kind of check through those options just to make sure they are correct. And we can give it a bit of a demonstration here, and you can see how it performs across the different taxa. So as I mentioned, this is a Pacific gull, and this is a nice, useful case because this is actually a juvenile Pacific gull, which looks quite different to an adult. But we can see that when we test the computer vision, it actually gets it straight away. So you can see they click in the bar, and the top option is Pacific gull. And you can see the adult looks very different there with white and dark plumage. So there, it nails it straight away. We can test it with this plant as well. This is Oxalis glabra. We can check there, see what it suggests. Oxalis glabra is the very first option it suggests. Again, it got it perfectly correct. This one, this caterpillar here, this is in the genus Pterocellus. You can see what it says. Pterocellura, boom, got it correct as well. But here we throw it a bit of a curveball. So this is a very obscure rare land snail that I found up in the desert in Western Australia. This is False Pleuroxia overlandarensis. Let's see what happens when we test the computer vision on it. You can see that none of these options are correct. Most of these species don't even occur in Australia. Some of these aren't even snails. There's a couple of fungi that are suggested here. So all it's trying to do is it's trying to find a visual match. It can't find a perfect visual match, so it just suggests a whole lot of incorrect options. So this is a case where we would have to manually enter the name. So this is false Pleuroxia overlanderensis. So just to reiterate that point, if you do test the computer vision, again, that's completely fine, but just if you do get presented with a few options where you're not quite sure about them, it is always handy just to check each of those options and see whether, first of all, whether they even occur in Australia, but also whether they do actually look like what your organism is. If you do happen to select a computer vision option that is wrong, it's not the end of the world. So it's not a big deal. Someone will come along and help correct that identification for you. But I always find it good practice to improve my own identification skills just to check those options as well before I submit them. So that's pretty much all the observation process entails. It's pretty simple once you know those basic rules and etiquettes. It's really simple, very, very streamlined. The other reason that as well, I'll mention that I always recommend using the desktop version to upload your photos is that if you're on the phone, you can only upload one observation at a time. So if you've gone out and you've photographed 50 or 100 different things, it's a pretty tedious process to go through and upload those manually one by one. Whereas if I'm on the desktop, there really is no limit beyond the performance limits of your computer of how many observations you can upload simultaneously. I will regularly upload anywhere between 100 and 150 observations at a single time. So it's just a much more efficient use of your time to upload things via the desktop. But if you are out on the go and you aren't taking photos of many things and you're just doing it for the convenience, then by all means, you can definitely use the phone app. And it's a relatively similar process to here. And all the same rules apply. Multiple photos of the one thing, one observation, photos of different things, different observations, and you've got to fill out those same 3 data fields as well. So it's pretty similar. So once you're happy that you've got everything filled out, all you would do is go up and you'd click this green Submit Observations button, and your observations will be posted to iNaturalist. So from there, you'll be taken back to your home screen, which looks like this. You'll be able to go to, if you want to check your observations at any time, say that you wanted to go back and double check something that you saw, or suddenly you realized an identification for one of your things and you want to go back and update that identification. All you have to do is go to your observations at the top of the screen here, and this will show you everything that you have uploaded to the site. So it's arranged in descending chronological order. So the top left will be your most recent observation, and then going down, the observations will gradually become older and older. So this is the observations, the grid view. You can also look at a map view. So you can see exactly where on the map you've made all the observations. So you can see here, I've made a lot of observations along the New South Wales coast, but I've also done big trips to WA, out to outback New South Wales, Heron Island, Northern Territory as well. You can also see things as a list, which gives you a little bit more details for each observation. It shows you the time and date, the place, and so on. A really important thing as well with iNaturalist is to make sure you respond to your notifications. So these function in exactly the same way as they would on any form of social media that you'd use. Anytime someone comments on one of your observations or you get tagged, so you'll have a username when you're using iNaturalist. So my username is the Beachcomber. And if someone wanted to contact me, then all they would do is type in the @ symbol and then the Beachcomber, just like you would on something like Twitter or Instagram. And then I'll get a notification. So your notifications will appear as a red speech bubble. So up here, you can see in the top right-hand corner, I've got one notification at the moment. And it's always really important to check and respond to those notifications, because often they're notifications from identifiers that are trying to help identify your observations. So this might be a comment saying, asking for clarification. So if you took a photo of a butterfly, they might say, what plant is this that you photographed it on? Or do you have any extra photos from other angles? Or are you able to go back and take a better photo of this feature so that I can help you ID? So it's always worth checking your notifications. So you can click there and you can go ahead and you can go to that observation. So this is an observation that I had previously interacted with, and now someone has added an extra ID, and you can see it's just updating me on the content. So anytime any content that you have interacted with in any way gets updated in some way, so a new ID gets added, a new comment gets added, you'll get a notification for that. Now, you can also explore observations from other users on iNaturalist, not just your own observations. So one of the really fantastic things about iNaturalist is you can use it as a way to find organisms that you want to see yourself. So say that you really wanted to see a particular species, well, all you have to do is go and explore other people's data, and you can see exactly where they saw one. So let's say, for example, that I wanted to see a waratah. I know that they're flowering right now, and I was very keen on seeing a waratah somewhere in Sydney, but I didn't know exactly where to go and find one. So all I'd have to do is go to the search bar here. I can type in waratah. We can go to this option here, which is the Sydney species. I can click View Observations. Now I'm looking at all observations of waratahs that have been submitted on iNaturalist. So you can see there's almost 900. So here I can swap to the map view, and I can see exactly where waratahs are being submitted for Sydney. And I can zoom in and I can say, okay, let's see if there are any that are in my area. And you can see really there's kind of three main clusters around Greater Sydney. You can see there's a big cluster around Ku-ring-gai National Park, big cluster around the Blue Mountains, big cluster around Royal National Park. And then I can actually zoom in further and keep going in closer, and I can see the exact points on the map where all of these observations of waratahs were made. I can click on any one of them, and I can query this individual record just by clicking on it. And I can see exactly when this observation was uploaded, who made it, where it is, and all these other kinds of interesting metadata. So this is just this really useful way of saying, well, there's this particular species or species that I want to see. I don't know where to find it. You hop on iNaturalist and you can see exactly when and where it's been uploaded. What's also really useful about iNaturalist is that you get life history and phenology charts that get produced. And I'll show you what I mean about that. So of course, I could go out and see waratahs, but I might not be able to see them flowering, right? So if I obviously want to go out and see a waratah, but I definitely want to see the flower. There's no point in me going out and just seeing a bunch of leaves, right? So what I want to do is not only am I checking where in Sydney that I can see waratahs, but I also want to check exactly when they'll be flowering so that I can go out at the same time. So you can see if I scroll down on this page here, so I should just note, I'll put it in the search again. If I type in waratah, whereas previously I clicked View Observations to go and see all the individual records, this time I'll click About, and that takes me to the home page for that species. If I can scroll down, we can see here there's a nice little chart. And if we go to plant phenology, you can see exactly when things have been annotated, and you can see the red here is flowering. And this is all the ones that have been annotated as flowering. So you can see pretty much if you want to see a waratah flowering, you should probably go somewhere between September and October, because that's the peak of the flowering period. You can probably get away with things if you went in August or November, that there are going to be some flowering. But generally, to maximize my chances, October and November. So another really handy tool for going out and finding species yourself. And you can explore any part of the world and explore any species. So you can just click the Explore tab at the top of the screen here. This will take you to all observations submitted across the world. You can then filter this down by place or group of organisms. So I can actually filter this down to say Willoughby Council. Whoops. So I'm now looking at all observations that have been submitted for the general Willoughby Council area. So you can see there's almost 4,000 contributed by 400 observers, and you've got almost 1,000 species that have been recorded in your area. So again, we can swap to the map view, and you can see exactly where these observations have been submitted. And so again, that's a really nice way of going out, and if this is your local area, of finding exactly where people are seeing certain groups of organisms. And you can filter this down to anything you like. So maybe you're interested in butterflies. So you can filter it to butterflies. And you can see that 14 different species of butterfly have been observed in the Willoughby area on iNaturalist thus far. You can swap to the species tab and you can see exactly which species those are. You could swap to something like mammals. And you can see there have actually been 10 different mammal species observed in the area. So this ranges from everything to flying foxes, to echidnas, possums, bandicoots, and wallabies. So the options are endless. There are 1,001 different ways that you can explore iNaturalist and use the data as well, as not only just observing, but you can also identify things for other people, and you can use the data in research. So I thought I'd just round things out by just briefly talking about best practices for photographs before I take any questions from people. Something that I alluded to earlier is that whenever you are going out and making observations, it is always best to take as many photographs as you can. Now, this will obviously vary depending on what you're photographing. If you're photographing something like a magpie or a cockatoo, then typically you'll probably only need one photograph and you'll be able to identify that organism from just that single photo. But more often than not, for things like plants or insects or snails or spiders, you might have to take multiple photos in order for it to get identified. A really common thing that you see from new users is that they might take a photograph of a eucalypt from 50 meters away and then upload it and ask for it to get identified. But in most cases, that's going to be impossible. It's just not possible to identify that group of plants from just that one photo. So if I'm taking a photograph of a eucalypt tree, I'll take a photograph of the whole tree, but then I'll take a photo of the bark, the leaves from both sides, the fruit, the flowers, the buds. So there's all these different characters that you have to try and capture in order to identify them. So as a nice general rule, and again, it varies even within plants, but as a nice general rule, if you're photographing plants, I always try to photograph the bark, the leaves, both a leaf from above and below, the stems, if there are any hairs on them, because hairs can often be important for identification, fruits or flowers, if they have them, a photograph of an entire branch, just to show how the leaves are arranged on it, and then, of course, a photograph of the whole plant. So you can then see whether it's a tree, a shrub, a climber, or so on and so forth. If you're taking photos of things like insects and other invertebrates, always best to try and get a photo from above, from the side, and from in front if you can. If it's safe to handle, so if it's something like a Christmas beetle, for example, and then you might like to pick it up and get photographs from underneath and behind as well, because these can also often be useful. But of course, don't handle things like spiders and snakes and things like that. But just, yeah, as a general rule, try and get as many photographs as you can, because the more photographs you have, generally, the more information is in those photos and the easier it makes it for the people identifying your observations. So I will wrap things up there so that I don't end up talking for another several hours about iNaturalist, and I'm more than happy to take questions from people.
Well, thank you so much. That was really informative, and hopefully now we've got all the information that we need to get outside and start making observations. So we do have one question in the chat from Marky. I think she's asking about the metadata on the photos that you upload. So she's asking if the timestamp is based on the location.
Yes, it is. So whenever you took that photo, then the timestamp should be associated with when and where you took the photo. So yeah, that's a good question. Sometimes you'll get someone say they'll collect a seashell from the beach, and then they'll take it home, and then they photograph it at home. The location and the time shouldn't be where you took the photo. It should be where you first collected that shell in that case. So the time will be based on the location where you saw that organism.
Awesome. I do have one question. So I noticed that you, correct me if I'm wrong, I noticed that you had a photo of what appeared to be a dead snake when you were showing us the notification that you got. So does that mean you can upload photos of, animal remains?
Yeah, absolutely. So not just photos of living organisms, but on iNaturalist, you can upload any photograph of any trace or evidence of an organism. So that includes feathers that you find, footprints, poop, dead animals, you name it. If it's evidence that an organism was present there, it is completely fair game. And a lot of times uploading observations of dead animals or bones or remains or things like that can be really useful for researchers. So there's even a specific sub-project within iNaturalist that explicitly records road-killed animals. So if you happen to come across a wombat or a kangaroo or a bird that's been hit by a car, you can take a photograph of that, upload it to iNaturalist, and it'll go into this specific roadkill project. And there's actually a number of researchers looking at that data right now, trying to understand which kind of species get hit by cars more often, where are the hotspots for getting hit by cars, and strategies we can implement to reduce these kills.
And so is there somewhere on iNaturalist where you can type in that sort of information saying it is a scat or a feather, etc.?
Yeah. So there definitely is. So when you have uploaded your observation and you go to that individual observation, on the right-hand side of the screen on the desktop, there's a menu called annotations. And under those annotations, it's exactly the kind of things you're talking about. So you can specify whether the thing you saw was male or female. You can specify whether it was dead, alive, or uncertain. And then you can also specify what you actually saw in terms of the evidence. So was it the organism itself, or was it a footprint, scat, bone, so on and so forth. So all that's like an additional layer of metadata that isn't necessary for your observations, but it is that nice extra level of information that you can add afterwards if you want.
Okay. Does anyone else have any other questions? Feel free to pop it in the chat. I do have one more question. Do you mind sharing with us maybe some of your most exciting or most memorable or even rare finds?
Yeah, absolutely. So a really cool recent one, and I like to tell this one because it really highlights that it doesn't matter where you live. Even if you're in a very, very urban area, you can still find really exciting organisms and make meaningful discoveries as well. So over the past 2.5 years, I've spent about 365 hours surveying a bushland reserve near my house out in Western Sydney. And I'm about to publish the report based on that survey. And I live in a very, very urban area of Sydney. A lot of industrial complexes, commercial estates around golf course, major roads, sporting fields. So the remnant bushland that is there is quite degraded, and it's surrounded on all sides by a lot of human land use. So you would typically expect there to be not much around. But in just an area of about 24 hectares, over the last 2.5 years, I've found 1,900 species in this small single bushland reserve near my house. And in particular, last year in March, I was going through the leaf litter and bark, and I found this interesting looking silverfish. And it was completely white, and I didn't recognize it at all from others that I'd observed before. So I took a photo, uploaded it to iNaturalist. An entomologist from Austria identified it to family for me. Then, because I knew the family, I was able to contact the Australian expert in that family. He said that looks like something brand new and undiscovered. So I went back a couple of days later. I managed to find another one of these silverfishes. I collected it. I mailed it to the expert. Turns out it was a brand new, never before seen, undescribed species of silverfish. We've now described that species. And so that is now a brand new species of insect that was just in the middle of urban Sydney. So it's if you can go into your backyard, if you've got a backyard or a front yard, there are almost certainly a handful, if not more, undescribed species and other invertebrates, just literally in your backyard, no matter where you live. So whether these are tiny spiders, microscopic flies, springtails in the soil, it doesn't matter where you live. There's all this amazing biodiversity around you if you really know where to look and what to look for. So even if you live in an urban area, it's really, really great to get out there and start looking, because you'll be really amazed at what biodiversity you'll actually find.
Does that mean you get to name that particular species?
Yes, I was an
Okay.
author on the paper. Yeah, so that was pretty exciting.
Yeah, so what did you end up naming it?
So that one was called Subtrinomura epigea. So a bit of a mouthful, but it was the epigea part is that it's typically, so it's found on the surface, this particular silverfish, which was quite unusual for that group of silverfishes. Normally they're found in caves and deep underground, whereas I found this one on the top of the soil.
That's very, very cool. I guess it just goes to show how important it is for us to start making observations and just start paying attention to small details, and how important it is to preserve all the remaining bushland that we have, especially in the city. You know, in the Sydney region, we do have lots of small pockets of bushland, and even though they are surrounded by urban areas, there's so much life in there, and if you just start paying attention, you'll start to notice them. Kathy is asking if we will get a copy of the recording. Yes, so we are recording this webinar, and I agree there was a lot of information there, really, really useful information. So I will be sending out a link to this recording so that you can always go back to it. Like Thomas mentioned, the Great Southern BioBlitz is happening very shortly, between the 28th to the 31st of October. So I do encourage you to use this newly acquired knowledge to go out there, even just to your own backyard, to start recording. Also, we do have a mock BioBlitz next weekend as well, at Watergate Reserve, which is not too far from most of you, I believe. So I'll be sending out details regarding that activity as well, if you're interested in joining us. So yeah, if there is no more question from the audience, I think we will leave it at that. But thank you so much, Thomas. That was wonderful. I think we all learned so much, and I'm now very inspired to go outside and start recording. And hopefully I can also be like you, discover a new species. That sounds very, very cool. So yeah, once again, thank you so much, and yeah, see you soon. Thanks, everyone.